Standardization and comparison between alternative techniques for isolation of microvesicles from plasma samples, avoiding lipoprotein contamination. My name is Laura, I'm a master's student, and my advisor is Professor Eugênio Damasceno Rotz. Microvesicles or microparticles are a type of extracellular vesicles with a size ranging from 100 to 1000 nanometers, formed from evaginations of the cell plasma membrane. They can be secreted by different types of cells, especially when cells are activated or apoptotic through mechanisms not yet fully elucidated. The content of MVs is quite varied, comprising different biomolecules from their parent cell. And the MVs are able to transport and deliver their content to target cells, therefore being able to actively modulate a variety of cellular events and participate in cellular communication. Currently, the interest in research involving microvesicles and their role in physiological and pathological processes is growing exponentially. However, the absence of a gold standard for techniques used to isolate MVs from biofluids results in reproducibility and contamination problems. MVs obtained from plasma have lipoproteins as one of the main contaminants because, in addition to the significantly higher amount of lipoproteins in plasma, MVs and lipoproteins share an overlap in their density and diameter spectrums. Plasma lipoproteins are spherical biomolecular complexes that have a central core of apolar lipids, surrounded by a monolayer of amphihepatic lipids, to which apolipoproteins are associated. Lipoproteins are divided into different classes according to their density and content. Plasma lipoproteins, besides acting as crucial mediators in the uptake of circulating lipids, also has a wide participation in biological processes. Thus, the need to search for alternatives for the isolation of MVs without contamination by these molecules is undeniable, especially in assays that seek to evaluate the functional characteristics of MVs, since the results can be biased by the presence of lipoproteins. So, the aim of this project is to standardize a protocol for the concomitant isolation of microvesicles and lipoproteins from clinical samples, avoiding possible contamination. We will work with two methods, the density gradient ultracentrifugation and the size exclusion column, which will be evaluated separately and when combined. The fractions obtained after each procedure will be then analyzed through colorimetric assays and flow cytometry to identify the samples containing lipoproteins and MVs. Then, the MVs will be quantified in the samples and their cellular origin will be investigated. In future experiments, we intend to assess positioning of exosomes and analyze the integrity of isolated vesicles through electron microscopy. Since a part of the MVs population may not express phosphatidylserine, we will also evaluate the expression of other markers. And finally, we will investigate function and profile of isolated MVs in infectious disease.